Well, thank you very much for the introduction, and thank you everyone for staying, and I'm really happy to be back again. And this is just a thank you, Esper. I found this really nice picture online of you, and I wanted to thank you again for hosting such a phenomenal conference, and it's really a pleasure to share with you some of the work that we do in our lab. So our lab in Hawaii is focused mostly on studies related to comorbidities. I think you've had a lot of interesting overview of the effectiveness of antiretroviral therapy. We are in an era now where a number of comorbidities are still persisting despite fully suppressive antiretroviral therapy. And comorbidities is an area that we work on. We also work on cure-related strategies. And hopefully, if I get invited back, I'll tell you more about our cure studies. But today I'm going to talk to you about the complications of HIV that are persisting in people who have been on effective antiretroviral therapy. And I hope to tell you some of the studies we're doing to try to see what we can do to mitigate this complication. And I think really this just highlights the success of antiretroviral therapy. You can see here the number of people who die in the HIV negative group right here is relatively stable, but you can see with HIV over the years the numbers have steadily declined in death rates, but they still haven't closed the gap between the HIV negative population. You can also see here on the right life expectancy. On the closed circles, you can see in the HIV positive group, sort of up around here, while in the HIV negative group, you can see in the open circles, again, we really failed to close the gap of life expectancy with the HIV population, sort of a little bit lower than what you see in the HIV negative groups. And really, we know that half of the deaths, at least in the United States, and around the world, and now if you have good therapy, are really related to a number of non-AIDS related comorbidities. As you can see here, AIDS-related deaths are still important, but we're seeing a much greater representation of different causes of non-AIDS related deaths. Again, I think in the early slides over the course, you've heard about malignancies and cardiovascular disease, liver disease, and non-AIDS infections really taking a greater representation of the causes of death. Again, a lot of this is really, we believe, due to ongoing inflammation that's not relieved by antiretroviral therapy and, of course, other comorbidities. Our interest in our lab has really been focused on the brain. We think this is another organ that is not restored to normal. There's a lot of data now that even despite antiretroviral therapy, we still see persistence of cognitive abnormalities. I'm going to talk to you a little bit about this specific organ. I don't think there's been a lot of discussion about this at this meeting, and I thought this would be something different from other organs such as the heart that have been discussed about earlier today in metabolic disease. So my talk will really give you a little bit of background, and I'll show you some insight of some of the work we're doing with HIV persistence in the brain and cognitive deficits that we see, new strategies we're using to try to look for biomarkers that I think are important in understanding the pathogenesis of cognitive impairment and brain abnormalities, and then some therapies that we're trying to use to try to mitigate these comorbidities and some future directions that are moving forward in the field. And as shown earlier, I'll try to stratify a little bit of Portuguese to help you follow the talk. But we'll look at, if you have good viral suppression, we still, with a number of neurocognitive tests, still can identify about 40 to 60% of people have some sort of neurocognitive disorder. And this could be attributed to a number of features in the CNS related to neurotoxins and neuroinflammation. So we're really interested in identifying biomarkers that could be important to understand the biology of cognitive impairment, and hope this will be useful in developing therapies to try to reduce these outcomes. And if you look in the pre-ART era, you could see that HIV really was a disease of the brain. There was a lot of dementia and sort of milder forms of neurocognitive disorder, and these can be picked up by neuropsychological test performance, some asymptomatic neuroimpairment, and in L is the normal cognitive incidence. But as we went into the post-heart era, we began to see a larger increase in the representation of milder forms of impairment. There's a reduction in dementia, but we still do see these deficits. And when we try to describe to people what are we talking about when we talk about brain abnormalities, they really are stemming in three great areas of cognition, behavior, but also motor impairments. And you can see sort of the features that occur even though people have been virally suppressed for a long time, we're able to pick up memory loss, concentration deficits, a little bit of mental slowing. The behavior with apathy and depression are also very significant in this population. But also we do see motor deficits that are very different from what you see in the HIV negative population. Right now, there really is nothing that we can do in terms of therapies for cognitive impairment in the HIV population. But also, when you think about HIV impairment, and I think we've had a number of talks about additional comorbidities that are related to impairment, it's always important to realize that alcohol, substance abuse, co-infection, nutrition, accelerated atherosclerosis, these are all cofactors that can be profoundly important in these deficits, including traumatic brain injury, sleep disturbances, but also several psychiatric illnesses are all encompassed within these age-related events in the brain. And so it's important to take this into consideration. When we think about the cells in the central nervous system that are vulnerable to HIV infection, a lot of the data now indicates that the myeloid populations, the macrophages and the microglia are really essential. They have CD4 receptors, they have the chemokines that are involved in viral entry, they're susceptible to HIV, and there is data that I'll show you suggesting that perhaps they could also be sites for reservoir persistence. And this nice review sort of talks about the selectivity. What we've been interested really has been in the myeloid cells in the blood, the monocytes. If you look at the old nomenclature that was described by Ziegler back in 2010, we identify about 3 populations of cells that circulate. The majority of the monocytes are classical. They have a lot of CD14 on their surface. They lack the CD16 Fc gamma receptor. And the inflammatory monocytes, which are intermediate in the description, tend to be more inflammatory. They express a little bit of CD16, and the non-classicals, these are very few in the circulating cells in the blood, tend to have a higher representation of CD16 on the surface, and there's a belief that these monocytes are more likely patrolling into the tissues and around the body. So as I mentioned, there are a number of chemokine receptors that are expressed on these cells, and there's been some data suggesting that they're related to cognitive impairment. Here we've shown that if you block with a CCR5 agonist, a couple of papers have now sort of shown some impact, at least in improving cognition. The mechanisms are still unclear. We know that there are other receptors on the monocytes, such as the 163 molecule, that gets cleaved, and the soluble levels of soluble 163 are elevated in patients in the blood, where you can actually use this marker as a biomarker for cognitive impairment in HIV. So, we think the monocytes in circulation may be an important parameter that has been linked to brain abnormalities through a number of these studies. And the hypothesis really that has been in the neuro-AIDS field has been that because our antiretroviral therapy is effective in suppressing virus, it really is inadequate in controlling persistence, and we think that could be occurring at least in the cells in the blood, that there is still persistence of virus. There is belief that these inadequate effectiveness of antiretroviral therapy, at least at the cellular level, allows these infected monocytes to persist. We think they're able to traffic through the blood-brain barrier to enter into the CNS, and at least this could be one way by which the virus may enter into the CNS. And then there's a belief that these monocytes may actually convert or transverse virus into these perivascular macrophages and really seed virus into the brain. There is belief that there's a pro-inflammatory environment that leads to neuronal injury and dysfunction, which eventually can be presented as cognitive impairment when you do a number of tests. And just for translation purposes, we really believe that this could be an important medium by which virus may enter into the brain. But there's been some very exciting developments in the last few years, and a couple of papers in Nature just came out, and in JAM recently, and a recent paper in eLife showing that there could potentially be a lymphatic vascular system by which virus, or at least by which the brain communicates with the lymphatic system. And this has now been shown not only in mice, but also there have been some human studies demonstrating the existence potentially of this CNS lymphatic vessel system. And this has now been shown in some new studies with non-human primates as well, showing that the meninges perhaps may harbor lymphatic vessels, and you can actually visualize this with MRI non-invasively, and I think this has developed tremendous interest in the neuro-AIDS field as another route perhaps by which HIV can enter. So we wrote whether this could be a trajectory for HIV. We wrote a review to try to understand whether this particular route may be an entry point. And using newer technologies, I think Tim Schacker has spoken about earlier, of RNA-scope, which allows you to look for virus. And these are autopsy specimens of individuals who had died on ART. You can actually see very clear P24 staining within the meninges, and by RNA-scope, at least in the cerebellum, we can actually identify 163 positive cells actually having HIV RNA by this methodology. So we think there's a different porous route other than the blood-brain barrier by which there could be potential entry of virus into the CNS, and this is something that we're particularly interested in pursuing. So in the area of how could we try to identify biomarkers that could be useful for identifying cognitive impairment, the field is really focused on a number of techniques to try to identify features of CNS injury. Neuroimaging has been an important area to look at metabolites in the brain. DTI and functional MRI have been used. Recently there's some work with microRNAs. I think Hendricks today spoke about a number of soluble markers you can look at in the blood, and sort of neopterin, a marker of macrophage activation, SB100, which is a marker of neuronal astrocyte injury, have been used. A number of markers of Alzheimer's disease have also been looked at potentially as markers of neurocognitive impairment in the blood, but also in the CSF. Immune activation, I think you've heard a lot about these markers being important and as drivers or persistence that persist despite antiretroviral therapy. And recently some work with lipids have also come about as potential markers of inflammation. But Michael Corley in our group, he's just recently joined the faculty, has we've been working together about trying to use epigenetics. This is a very interesting area that we think could be useful because epigenetics really is the study of mechanisms by which the chromosome is bound, and you get these heritable changes to the genes which really do not get impacted by the DNA sequence. And these processes now have been recognized as being important in regulating many, many genes. So just to give you an example, if you look at the genome, what we see is this epigenome are sort of the areas that regulate the genome, and there are two major routes by which epigenetics can be measured. One is DNA methylation. You have a methyl group that is added to a cytosine base here, and that really can regulate gene expression. Histone modifications, such as HIV, as you know, is sort of the accessibility of gets shut down by the virus as a way to allow persistence. And these sort of two methods have been shown to regulate gene transcription, and the modification of the accessibility and the compaction really can allow the genes to transcribe. So we've really been trying to take advantage of epigenetics as a way to understand biomarkers. In the oncology field, if you look at different tissue specimens, they've actually used DNA methylation as a way to help with diagnosis. You can actually distinguish in this array between tissues that are benign and malignant. There's good stratification. In this heat map, you can see very clear differences. Each line represents a different methyl site, CPG site on the genome, and each line going down is an individual patient, and you can see very clear differences. You can use them as a way to look at therapy responses. You can see these are individuals with good responses and a different methylation level on these CPG sites with somebody who has a poor response. You can also use them to identify the tumor types and also the progression of the disease between short-term survivors and long-term survivors. So a very powerful tool as a biomarker of oncology in the oncology field. So we sort of came up with the rationale that, given that monocytes are accepted as a important player in the pathogenesis of cognitive impairment, and there's a likelihood that there is some memory-like activity, at least driving different functional changes through epigenetics in the myeloid compartment, we wanted to try to determine whether we could find a signature as a biomarker using an array, whether we could look in the monocytes and see if there are any related changes in the DNA methylation profile. So we took a small cohort of individuals here within our aging cohort in Hawaii, And you can see here we had a group that were not impaired cognitively, and we determined that by using a composite score of neuropsychological test scores. Less than 0.5 tends to show impairment, so you can see in about 11 individuals there was impairment, while the other group were not impaired. Their scores were relatively high, and they were relatively equal in terms of age and CD4 T cell counts. And while some of them were viremic, all of them were on ART, but at different levels of viremia, but still there's no difference in viral load between the two groups. And what was very exciting was that by looking at the DNA methylation in a 450,000 methylation array, we were able to very nicely stratify these two groups by the level of DNA methylation. And you can see in this heat map, the greater the methylation, you can see in red, and in blue you can see less methylation at these individual sites. And you could actually very nicely stratify the impaired group in green compared to the group in brown who are not impaired. So very easy to stratify that. And we found about 1,000 sites in the genome, in the monocytes, that were actually differentially regulated by this array. So using this discovery cohort, we were really excited to find potentially new biomarkers that will be able to help us distinguish between these two groups. And this work has now been published as a new way to define an immunoepigenetic signature of HIV impairment. And these are just 3 genes. And what was interesting was these are 3 loci within 3 genes that have been associated with the central nervous system, bone-derived neurotropic factor. You can see here the groups that were impaired had a lower methylation level compared to the groups that were not impaired at a different sites within the genome, within the promoter, but also within the gene bodies, as you can see, or the intergenic regions. And these 3 sites have been shown to be related to the CNS, and this we were able to pick up within the monocyte population. Another set of genes that sites that we observed, this is one particular gene, the thrombospondin 1. You can see these 3 sites within the promoter region were also differentially methylated. As you can see, the impaired group had lower methylation levels. And what we did was to try to see whether this was related to transcription. So we looked at expression, and you can see there was a very clear demonstration that the lower the level of methylation, the greater the amount of transcription of this particular site. And you can see that this particular gene has been shown to mediate cell-to-cell interactions, but it's also been shown to potential biomarker of plasma viremia in HIV patients. So again, here we're showing that within the monocytes you can separate, at least by looking at a couple of these methylation sites, genes that are involved both in the CNS but also with HIV itself. And when we performed a number of neuropsychological test batteries within these same individuals, these allow us to measure through a composite score the behavior, the attention span, working memory, a couple of visual and verbal learning parameters, but also motor assessments in these individuals. We can actually get a composite score of their neuropsychological test performance. And when we correlated these with a number of these methylation sites, we found very strong associations between several sites within the genome that would stratify their impairment with a level of methylation. So 2 separate independent measures of impairment were linked to levels of methylation within this cohort, this discovery cohort. So, just to summarize, we thought this really is the first evidence of a distinct immunoepigenetic signature in monocytes that would help us stratify cognitive impairment. These DNA methylation signatures, as I pointed out, have been linked to the CNS, but also sites that are interacting with HIV as well. And finally, we think that these differences in methylation are associated with gene expression and also with these neuropsychological test scores that we're able to put together. So, this is all very exciting. This cohort, as a discovery cohort, had a variation in viremia. There was also variation in antiretroviral therapy use. So what we did, and this is new data that we are sort of working on right now, was just go into more detail on some of these particular sites. And one of the sites that we looked at that was very strongly segregating the 2 groups was the IRF8. This is interferon regulating factor. This is a member of the IRF proteins, and it's a transcriptional regulator of the interferon system. And it's involved in pathogen-associated molecular patterns and TLR signaling as well. So this particular molecule, as you can see, the loci that we identified in the genome, there were a couple of them that were different, but this particular site, you can see by when we looked at expression, we could see a very clear association with the greater methylation in the group that had impairment was related to greater protein expression. So this methylation at this CpG locus in the promoter region of the IRF8 was significantly higher in the monocytes from the group that were impaired versus the group that was not impaired. And what we did was to sort of look at what IRF has been shown in the literature. You can see that in at least the monocyte cytokine response to pathogen stimuli is really regulated by IRF8. You can see interferon beta secretion when you add a virus and you transfect with IRF8. You can see you can actually increase the level of interferon beta production with IRF8 transcription. What's also very exciting is that it's actually involved in myeloid development. If you actually mutate the IRF8 gene, you actually get a complete depletion in both monocytes, actually, in these three individual patients, a mother and the father and the progeny. You can actually see the IRF8 mutation depletes not only the myeloid cells, but also the dendritic cells, which we actually found some interesting data on that. So there is some role for IRF8 in myeloid development. But also, it's actually been shown to inhibit HIV transcription. You can see here in this data set, when Jurkat cells that are infected with HIV, and you actually stably introduce IRF8 into this vector, you actually get a complete shutdown in HIV P24 production. So I think this is very interesting that IRF8 can also potentially regulate viral persistence. In fact, there is data showing that it actually has been involved in latency. It's actually, it also has the ability to block the activity through IRF1, so it's a dominant regulator of transcription. You can see this here with IRF1 being an intermediary. But also, it also can allow the cells to maintain quiescence with proviral DNA in this pro-monocytic cell line. When you have IRF8 gene expression being reduced, you can actually see, in sort of the dotted line, you actually see an increase in GAG mRNA expression. So really decreased IRF expression following latency reactivation suggests that it may be involved in maintaining viral quiescence. So all this data, we've picked up IRF8 through epigenetic profiling, and there is data in the literature suggesting that it's involved in both viral persistence but also in myeloid development. So we thought, well, let's just take the epigenetic data and see if we can see protein expression in a completely different cohort and see if we can use the IRF8 signal that we've picked up to validate what we're seeing. So we were very fortunate. We have a separate cohort in Hawaii, the Hawaii Aging Cardiovascular Disease cohort. And in this cohort, these individuals are very unique because we have actually not only been looking at their cardiovascular assessments, but we've also performed a number of neurological CNS-related parameters in this separate cohort. And this cohort is very interesting because everybody is completely virally suppressed. They've actually been virally suppressed for over 13 years, so very well suppressed. They've been infected for over 15 years. They have pretty good CD4 counts, above most of them above 500. So they're all doing pretty well. They're generally in the elderly age group, about 51, mostly male, which is representative of our demographics in Hawaii. But we also had a 14 composite score of the neuropsychological assessments done in this population. So completely separate population. But a very well-characterized cohort with neurological assessments. So we've also done a number of things. We've also done 1.5T MRI assessments to measure regional brain volumes in all these patients. And this subgroup of study participants within our cohort, we also have been able to measure a number of subdomains of, as I mentioned, of neuropsychological test performance, but also brain volumes. So by flow cytometry, we've been able to, I don't know what's happened to slides are disappearing. Okay. Anyway, for some reason I'm losing some slides. But we basically were able to show that we were able to identify IRF8 expression within the monocytes, and we found that there was a very good correlation between the absence of IRF8 in this population and worsening cognitive performance. So it does look like the IRF8 deficiency has been linked to impaired cognitive function in this population. And this was across many domains, both globally, but also within the learning memory, working memory, and executive function domains. In both the non-classical monocytes, but also in the classical monocytes, but also in the intermediate monocytes. So we do find an inverse relationship with impairment and the expression of IRF8 intracellularly within the monocyte population in a separate cohort. And we also looked at brain volumes. So we also noticed that those with smaller brain volumes measured by the MRI data sets were also associated with a reduction in IRF8 expression. So here we're showing within protein expression, we're able to almost validate that, at least in the myeloid compartment, there's a strong link between IRF8 expression and brain abnormalities. And so in a completely separate cohort, we can show that there is an important role for IRF8 transcription as a biomarker, but also potentially involved in the pathogenesis of HIV abnormalities. And it may also be a useful target that we are trying to look at that may be used to ameliorate cognitive impairment, but also HIV persistence. So one of the questions we wanted to ask was, these are all chronically infected individuals. These epigenetic imprints seem to have been imprinted in chronic infection, seem to persist and be related to impairment, but we wanted to know just how early these events of methylation are occurring, but also if we may be able to link any changes that may occur with antiretroviral therapy, with whether this was linked in any way to dysregulated immunity, but also inflammation and compromised host defense. So to do that, we turn to our studies that we're doing in Bangkok, Thailand, where we have access to the Search 10 RV254 studies, where we're able to capture individuals very, very early soon after infection. And you can see here, using a number of antibody and antigen detection kits, we're actually able to capture people within the first 30 to 40 days after infection. Using a number of algorithms, we can capture people very, very early after infection and try to use this data set with control individuals in, again, a different cohort and see if we can identify whether these epigenetic changes are occurring very, very early. The acute cohort is actually quite exciting. We're able to capture over right now we're at 500 people captured in acute infection. And the way we do that, within 2 days of diagnosis, and this is really by individuals coming every few weeks into the clinic, we're able to capture acute infected individuals. But one of the unique things about this cohort is within 1 day, these individuals are actually put on antiretroviral therapy immediately. And as for our neurological assessments, we're actually able to capture MRI, MRS imaging, and also to do lumbar puncture in these individuals. So we're able to capture some CNS measurements, and we've been able to do this in over 600 individuals so far. And this really allows us to use this acute cohort to study CNS abnormalities in the brain. So from this cohort, as I mentioned, myeloid activation is a really important driver of bad things in HIV patients. In fact, all-cause mortality, the 163 marker in the blood is a really good independent marker of mortality. You can see here with this stratified by different levels of 163. Those with the highest quartiles of 163 in the blood tend to do cumulative survival rates are actually much lower. So I mean, this sort of just shows you the importance of myeloid activation as a trigger, or at least a biomarker of abnormalities. When we look in our acute cohort, where we, as I mentioned, start treatment very, very early, you can see in chronically infected individuals, compared to controls, at least in the plasma, we can see that even despite 48 weeks of therapy in chronic infection, we still see a persistence of the 163 marker in the blood. In our acute cohort, what's really exciting is that really within Phoebig 1 and 2, which is in the first 14 days of infection, you can see that with early treatment, we really see very low levels of 163 very early. And with early treatment, we really are able to sustain and sort of keep those levels quite low. However, in Phoebig 3s, the levels already start to rise, and we're actually associating these with worse neurocognitive outcomes. However, with treatment over 48 weeks, we are able to suppress that, both in the blood but also in the CSF, as you can see here. So what we were able to do was we've now got a larger array looking at about 850 methylation sites. And we've been able to try to look for similar perturbations in the epigenome within individuals who are acutely infected within a few weeks, 17 days from infection, but also see what happens after antiretroviral therapy initiated immediately after diagnosis compared to a group of controls. And the supplement grant that we received is very exciting. We're actually able to show, at least for this array, that we're able to see that the very good stratification between the HIV negative groups compared to the HIV positive groups. There's very clear difference in methylation levels. But what was interesting was that even with early treatment, we really don't see much of a difference. There's actually an admix of the acutely infected individuals who received ART versus those that were just infected. Not much difference. So it does appear that this genome-wide changes that occur in the monocytes does occur very early, as you can see, but it doesn't seem to get impacted at all by antiretroviral therapy. So even though you start treatment very early, this very early epigenetic hit happens quite rapidly, and it's actually persistent and doesn't change. In fact, if you look at the HIV negative group versus the acute HIV positive groups before therapy, there's almost, when you look at out of the 850 sites, only about 28,000 change. But when you look at the acute HIV positive group with on ART, very few sites actually are differentially methylated. So it does appear that these changes happen early, and we're trying to work backwards now to try to see if any of the impairments or any CNS abnormalities that we see have really been set prior to ART as well. So I'm going to make a quick shift here to move to some of the therapies that we've been looking at for cognitive impairment. We're now trying to use a lot of our epigenetic data to try to build strategies that we can use to try to perturb those systems. But Michel D'Antoni in the lab has been very interested in some of the interventions that we've been doing. And again, this really stems from the chemokine receptors. We've shown some data that, at least in a single-arm study, that CCR5 inhibition may have an impact in not only in viral entry, but potentially in trafficking, but also in reducing inflammation in the brain. And this is one particular target that we've been looking at to block. We also know that the CCR2 molecule has been involved in migration. It's expressed on a number of monocyte populations. We know that MCP1 is an important biomarker of cognitive impairment, and we know that it's an important chemoattractant in recruiting myeloid cells into the tissue sites. And we've identified, at least in a recent paper, that the CCR2 molecule expressed on intermediate and activated monocytes does seem to be related to cognitive impairment. And what we know is that, at least in the brain, that there's potential role for cellular populations that express CCR2 for migrating into the brain and sort of getting involved in the cascade of chronic inflammation that potentially may drive neuronal injury and astrocyte dysfunction. So really we think targeting both CCR5 and CCR2 could be quite useful. And there is a drug, cenecrivarat, which is now being used. It's been used actually in a clinical trial to lower viremia soluble in HIV individuals on ART who are ART naive, and has shown some efficacy in lowering viremia. But it's actually being used now, or being looked at least in the setting of liver disease. It's been shown to have some anti-inflammatory and antifibrotic activity in animal models. And it is an oral drug that's able to block both CCR2 and CCR5. And we've been able to carry out a small single-arm study to see whether by blocking both these two chemokines, we may be able to improve cognition, but also decrease monocyte activation. And to do that, we looked at 17 individuals, and what was different from other studies is we actually tried to identify and screen individuals who had cognitive performance that was a little bit low, and we used a definition of the neuropsych scores at least having less than 0.5 in the composite scores, but also having single domains that were less than 0.5, negative 0.5 in their assessment. So we sort of screened individuals, identified people who were impaired at least by these parameters, and then carried out a 24-week study by administering the drug over time and sort of carrying out blood draws throughout this very short period. It's really just as a signal to see whether we could see changes in neuropsych testing, but also in markers of myeloid activation following cenecrivarat in this very well characterized pilot study of 17 individuals. And this is the clinical data. Again, these individuals have been infected for over 16 years. They've been 16 years on ART, but over 20 years of infection, but also very well suppressed, and as you can see, have pretty good CD4 counts, roughly above 500. Again, mostly male in this population. So one of the interesting things in the data set, when you look before and after 24 weeks of therapy, we did notice actually an improvement in the global domain scores. Was quite significant, but also in some of the subdomains of working memory and attention, but not much change in executive function or gross motor activity. And there was some sort of trends with psychomotor speed. So we do find a little bit of a signal here that this intensification of cenacrivirac may actually impact their cognition. And we looked at myeloid activation markers. These are all linked in some way to myeloid macrophage activation in the blood. And we do see a significant decline in all three of these markers after at least 24 weeks. So we think it's this combination of blocking CCR2 and 5 seems to impact myeloid activation, at least in the blood. When we did some correlative studies, we found one biomarker, neopterin, seemed to be associating with the changes in working memory. So essentially, the greater your change in neopterin levels, actually the better your performance in working memory actually was occurred. It did seem to predict, at least in a linear regression model, that decreases in neopterin over 24 weeks was associated with an increase in working memory capacity in this population. So again, a very interesting data set that I think we are now trying to move this into a double-blinded study to see if we can in a randomized double placebo-controlled study to see if we can use this drug as a way to perturb any impairment in our aging population. It's also just wanted to point out that this drug was actually well tolerated in this population. And then, so really just as I summarize for you, sort of how the field of neuro-AIDS has moved from the early stages of early description of cognitive impairment in a number of studies, sort of as I mentioned, beginning with the dementia complex that we identified in the early epidemic, to SIV models of encephalitis being developed, and the Charter study, which really allowed us to look at a larger population of patients with impairment to better understand the pathogenesis and better define criteria of impairment, to now understanding that inflammation is really a critical pathogenic element. And we are now trying to find ways to not only eradicate the virus, but also perturb impairment as we move forward and try to identify newer biomarkers that will help us in pathogenesis of cognitive impairment. And I sort of talked about our epigenetic studies that we hope will help move this forward. We now have a number of grants that we're using to try to perturb the myeloid reservoir. This is a new drug that we think has the ability to reactivate virus within the myeloid compartment. And we also have additional clinical trials where we're using Miravirac right now as an intensification regime. This study is fully enrolled, and we're completing the study in the next few months. And we have a new study in women where we're using a glucocorticoid intervention using this strategy in women, at least, some interesting mechanisms that we think could enhance cognition, but also lower inflammation. And hopefully, on other visits when I come back, hopefully I can show you some data on these two new RO1s that we're assessing interventions in HIV in patients with cognitive impairment. And then finally, looking at the CNS using animal models, we're really interested in sort of traversing these data sets into the non-human primate model. We have a new paper now that's just come out using a non-accelerated SI-SHIV model to try to understand CNS inflammation, but also persistence, and use these models for some of our interventions as well. And this sort of just gives you an understanding of the current models and viral clones that are being used to assess CNS, at least for encephalitis, and CNS penetration of virus into the brain. And we think this new model may be useful as a more replicative model of HIV to test some of these pathogenic questions, but also these interventions. So with that, I'll talk to you about how the myeloid field is moving forward in many new ways with newer technologies. This is a recent paper in Science showing that we can now identify beyond our 3 monocyte populations using a single-cell RNA-seq, at least in the blood, that we can identify newer populations of monocytes, actually. This is a 4th population of monocytes that can be identified using RNA-seq, but also identify new dendritic cell populations, which is sort of allowing us to interrogate newer populations as we go forward. And we're sort of looking at this now in different compartments as well as methods to sort of better characterize these myeloid populations in the setting of HIV. So with that, I really want to thank a lot of people who have been working with us on this project, mainly our colleagues at UCSF, at the Memory Aging Center. We have a new Cure initiative, the International Neuro-HIV Cure Consortium, which involves UCSF, Yale, and our colleagues in Thailand. And I really want to thank Tabira, who's been helping with the drug production, but also our funding sources through the NIH for these studies. So with that, I'm happy to take any questions from you. Thank you.
We'll be open to the question. Okay. I believe that you said twice that as soon we start the art, better for the brain, of course. Yeah. I want a question. If you believe that the IRF8, CCR2, and Elpterine will be good markers for you, we studied the damage of the brain all over the infectious disease.
IRF8?
Yeah.
Yeah, so I think, we weren't sure when we found the methylation differences whether this would be translated into a transcription. And using a separate cohort, now we can identify, at least in the myeloid cells, and I didn't show data, but even in dendritic cells, we do see very strong associations with the protein expression in these cells, and we think they could be potentially regulating not only the virus, but also inflammation at the cellular level. So this could be potentially a new pathway that may be associating with subsequent CNS abnormalities.
Okay.
Yes. Sorry.
Please.
Yes.
No.
Blood.
Yes.
Yeah. Monocytes in blood typically don't spend that much time there.
Yes.
And so you found these really sustained differences in their epigenetic modifications,
right,
that were durable despite long-term art. What do you know much about the environment in the marrow? Do you know much about what it might be in the blood that's irritating these cells?
I think you've hit the million-dollar question, which is we think there is some perturbation, at least because epigenetics is almost an imprint. It has to be something in the bone marrow. So you're right. We don't know quite yet what that trigger is, but because monocytes live for only 3-4 days, and you've got this continued almost epigenetic fingerprint that's perturbed, we don't know if it's set prior to infection or whether there is a bone marrow-derived signature of impairment or other diseases that regulating that transcription factor. But you're right. I think the bone marrow is the site to go to understand this persistence. But at least we now know that if we have someone with an IRF8 methylation profile, they're almost certainly going to be related to some sort of CNS perturbation. Yeah. Sorry.
A little further, do you know, have you or anybody else examined the progenitor cells of these precursor of myeloid precursor cells? Do we know anything
absolutely.
about that?
That's sort of the next stage where we're going. Yeah. And in fact, the difficult thing is convincing the clinical team to do bone marrow extractions, only because there's a belief that it's painful, but I think we might have to go back into that compartment.
Please.
Hi, this is good talk, and I'm very interested in the epigenetic analysis. And in the third part, you examine the effect of treatment. And are you going to examine the epigenetics, so methylations, after the treatment?
So your question is, are we going to look at epigenetic changes after treatment? So in the acute cohort, the reason we like that cohort was we are able to capture people just before they're infected, but also, they go on treatment the next day. So we have looked at the signature just after infection and after ART, and there's very little change. So we don't know what interventions may perturb that epigenetic imprint. So we're looking at a number of targets that we can see may be able to perturb.
But in the third part of your talk, you tried one drugs.
Sure. Yes.
And my question about that.
That drug. Yes. No. So we're actually going back now, and we're doing our epigenetics on all these different interventions. That's a great question. So we have another drug that we haven't finished unblinding. It's telmisartan. It's an angiotensin-converting enzyme inhibitor, and we're using that to lower inflammation, fibrosis. And in those specimens, we're interested to see whether there's any shift in the epigenetic profile and all these interventions. That's the hope. But we think we may need to also look at demethylating agents as well as more targeted therapies for methylation profiles to see if we can perturb that.
Thank you.
But great question.
Questions?
Good.
If you talk about the damage of the brain, have you seen something about the effervescence? Looking for the
yes, no, that was a good question. In fact, I'll just take you to the acute cohort data, if I can go back. The reason our acute cohort is interesting is we actually have a couple of stratifications now. So, half of them go on a mega heart regimen where we've included Miravirac, and another group where they're, stratified into ARTs. Now, these were efavirenz. Now we have a third group where we actually are using dolutegravir and integrase inhibitors. So we'll be able to tease out the drug effects as this cohort is advancing with therapies, and we're following them up. This was we now have almost 5 years of follow-up, so we'll be able to dichotomize whether the antiretroviral therapies will be, may impact the myeloid epigenetic profiles as well. But that's a good question. Yeah.
Thank you. Other questions, please?
Okay.
Okay. Thank you very much for your excellent